Assalamu alaikum students, welcome back to BioCore. So far we have understood what enzymes are, how they work, effect of temperature and pH. But now the question is, how do scientists use enzymes in real life? How do we perform enzyme activity in experiments? So today's lecture is very important because we will cover enzyme practical experiments from your book. Firstly, we'll discuss the safety precautions. Before starting experiment, safety is very important. Wear eye protection, handle hydrogen peroxide very carefully because it can burn the skin. And be careful when you are dealing with hot water or water baths. So we are going to start our first experiment, that is about the catalase activity from the living cells, and this is also known as the liver experiment. In this experiment we study an enzyme that is known as catalase. Catalase breaks down harmful hydrogen peroxides, and the reaction will be like this, that the hydrogen peroxide, when the enzyme acted on it, they change the reactants into the water and the oxygen. Now the enzyme that is using here is catalase, and the products are water and oxygen. Now it's time for discussing their procedure. First of all, we will grind livers with water and sand. This releases enzyme from cell. The first step is very important because to get enzymes from the cell. In the second step, we will filter the mixture because filtrate contains the enzymes. In the third is add filtrate to hydrogen peroxides because hydrogen peroxide is the reactant, and when the filtrate add on it, then the reactions occur. Now here are some observations, that bubbles form oxygen gas. Strong reactions, bubble oxygen can be testing using a glowing slit. There is the observing point that you have to observe in it from the bubbles. Boiling test. This test is also known as a boiling test. This is a very important concept in which we will boil the filtrate, then add it to hydrogen peroxide again. No reaction occur. Why? Because enzymes is denatured by heat. Yes, very good. Because enzyme is denatured by the heat. If we use this filtrate in a boiled condition, then the reaction will not be proceed. Now, there is a little bit comparison, that is with the catalyst. We compare this with catalyst, that is the manganese oxide, manganese 4 oxide. So this is the exact name of this. It also breaks hydrogen peroxide, but it is not an enzyme. Even after heating, it still works. Because it is not the enzymes. Enzymes are affected by heat. Catalysts are not affected by the heat. So this is the comparison, and this is the first experiment about the liver cells or catalase reaction. Now the second is that effect of temperature. Firstly, we will discuss a general catalase activity. Now we discuss the effect of temperature. For this reaction, we will use the enzyme amylase. So let's start study the temperature. So now let's study temperature using amylase enzyme. Amylase breaks starch into sugar, that is the maltose. Amylase is an enzyme that breaks down starch to sugar. Sugar name is maltose. Now first we have to make a setup for this preparations. Start solutions in tubes 1, 2, 3. We have to take the test tubes, that is 1, 2, 3, and also take a water bath. Like you can see here, we have taken the 3 test tubes that contains the starch 1, 2, 3, and in the next we take the 3 furthermore 3 test tubes that contains the amylase, and mention the name with A, B, C. So here there is a 6 test tubes. Now we have to take water bath. The first water bath is ice water, that's temperature is 10 degrees Celsius. Second is cool temperature, that temperature is 20 degrees Celsius. And third is warm temperature, that temperature is 35 degrees Celsius. So this is the procedure that we are going to perform this activity. Keep tubes in water baths for 5 minutes. Mix amylase with starch, add iodine solution. This test is known as the iodine test. Like in the previous experiment we do the boiling test, so today so in this experiment we will do iodine test. Iodine test means blue black. If blue black is present, color is appears, it means that starch is present. And if it's brown, so it means that the starch broken down. So this is the observations, that after mixing we have to find the colors. If this is the blue black color, this means the starch present, and if the test tube's color turned into brown, it means that the starch is broken down. Faster disappears of the blue color, faster enzyme activity. Warm temperature is the fastest, cold temperature is very slow, and very hot temperature if is this is also may stop and the enzyme will be denatured. So this is our second experiment in which we discuss the amylase activity and the effect of temperature. Now the third is also very important. In this experiment we will study the effect of pH. First we have to make a setup. In this experiment we will take the 5 test tubes with different pH. pH 9. pH 9s will be alkaline. pH 7 to 8, that is slightly alkaline. pH 6 to 7, that is almost neutral. And pH 6, that is slightly acidic. And pH 3, that is purely acidic. So these are the test tubes that we are going to take, and we will prepare the test tubes and manage the pH. Now there is a procedure that what we have to do. We take starch solution, add amylase. Okay. First we take the starch and then add amylase in it, and then add any acid or the alkali. Test with iodine every 30 seconds. If iodine stays brown, the starch digested and record time taken. Okay. Different pH value affect enzyme activity. Fastest reaction, the point at which the fastest reactions occur, it means that this pH is optimum. Too acidic or too alkaline, slow or no reactions. So this is all about the experiments that you have to perform practically. These experiments test your practical understandings, and the most important skill that you must have for your practicals is that measuring time, observing color change, comparing results, and the drawing conclusions. The most exam important point is that the catalase breaks hydrogen peroxide, heat denatures enzyme, temperature affects reaction rates, pH affects enzyme structures, and the iodine test is for starch. Let's quickly revise this. Enzymes can be tested experimentally. Catalase produce oxygen bubbles. Boiling stops enzymes activity. Temperature affects rate. pH affects enzymes efficiency. If you found this lecture helpful, like the video, comment your questions, subscribe to BioCore for complete biology lectures. In this next lecture, we will solve the past papers from the enzymes. Allah Hafiz.